All right, so welcome to a video lecture on astrocytes. There was previously a video that Eric made, which was a pretty long, dense video with a lot of advanced neuroscience topics. And a lot of the credit goes to Eric for this video as well, because this is just a more simplified and shorter version of what he produced. And we wanted to make this so that those of you who are just starting into the field of neuroscience, or getting a little interested, but you don't want to just jump straight into the cold water. Maybe you just want to get your toes wet. I think it's more appropriate to show you guys a simplified kind of entry-level video. But I still highly encourage you, especially those of you who are a little bit more advanced, to check out Eric's video, because a lot of the stuff that he showed there are really advanced neuroscience topics that you'll see in the university level and even in the graduate level. So if that's something that you want to do in your future, I'd highly recommend you watch that video and take detailed notes. Now, as for things that we could ask you for the Brain Bee, again, most of the questions, if not all the questions, are going to come from the Brain Facts book. We're making this series of lecture videos on glia just to add and supplement the stuff that you guys already have, to give you guys more resources in case you ever need to come back and look at this, if you ever need to in the university or in graduate studies or anything like that. So again, most of the credit for this video is going to Eric. I just kind of took his PowerPoint, modified it a little bit, and simplified it, kind of cut it short. So hopefully this doesn't take more than 15 minutes, I hope. So what are astrocytes? So astrocytes are a specialized glia. They outnumber neurons 5-to-1 in the central nervous system. And each astrocyte has its own domain of control in the central nervous system. So the astrocytes are going to support the neurons in a plethora of ways. Okay, so without astrocytes, neurons really wouldn't be able to function. So we know that neurons are quite sensitive in terms of cells. They need a lot of glucose, or at least a lot of fuel, whether it's glucose or ketones. They need a lot of fuel to function. And so they need to get that fuel from something. And we also know that not every single molecule or every single thing can pass through the blood-brain barrier. And astrocytes do play a role in the blood-brain barrier. And, without astrocytes, neurons wouldn't be able to eat glucose or consume glucose as well. And we would be prone to more infections and things like that. So astrocytes are super important. We're going to dive into some of those topics in this video. So here's some of the morphology. Different astrocytes in different areas are going to, or I should say, out of the 4 basic types of astrocytes, the morphology is going to differ a little bit. And morphology plays a huge role in the function of the cell itself. So I want you guys to take note of that. The shape and the pseudopodia of the astrocyte, the length of that, and you can think of the length of the feet. The size of the cell itself is going to play a huge role in how it functions. And we'll see that as that changes in some disease states, that you can actually hinder the function of the cell itself. So astrocytes have radially arranged processes, so they kind of look like stars. Radially arranged just means that around the cell body of the astrocyte, you're going to have appendages coming out, kind of like at every direction of the clock, basically. So they're very, very full of a bunch of feet. Okay, think of them as like octopi, okay, or like squids. And basically, the more arms and legs that it has, the more area that it can cover, which is good from a surface area standpoint, right? The more arms and legs you have, the more things you can touch and influence. Okay, so communication with the neurons and the vasculature and the interstitial volume, the extracellular matrix, all that plays a role, or all that is affected by the astrocyte. So astrocytes are also going to border gap junctions. So if you take a look at number 1 here, modulation of communication, astrocytes are present pretty much at every synapse here. So with the presynaptic and the postsynaptic neuron, there's always going to be an astrocyte foot there to uptake any excess neurotransmitter or influence the expression of certain things in the postsynaptic neuron. And they play a huge role in what happens in the pre and postsynaptic neuron, which is how we learn. So astrocytes play a huge role in that as well. You can also notice here, number 2, the astrocytes have, we'll say, 1 foot on the blood vessel. And this is really important because this is going to play a role in the tight junctions here, which are going to selectively limit what can pass through the vasculature into the brain itself. Okay. And then they're also going to support myelin coverage of the neurons. So again, they play a lot of vital roles. So here are the four main types. This is super important. So if you're going to focus on anything, try to get these down first. So there's protoplasmic, there is fibrous, there's interlaminar, and there are varicose astrocytes in the human brain. In different mammals, for example, rats, mice, they're going to have different number of types of astrocytes and probably less than we do. But we, as humans, have about four main types. There may be some subtypes, so we're going to focus on the four main types of astrocytes. And protoplasmic make up the majority of astrocytes. They're going to be very dense with a lot of radial processes. And they're going to be GFAP positive. So GFAP is glial fibrillary acidic protein, which is a characteristic protein that is expressed by astrocytes, especially adult and mature astrocytes. That's going to play a huge role when we look at some of the scientific studies in the end. Okay. Now, these are going to be found in the gray matter between layers 2 and 6. So if you didn't know, the cortex in humans have layers, and so we'll plan to make a video on that as well to thoroughly explain that. But for now, just understand that protoplasmic astrocytes can be found in certain layers of the cortex. Okay, the protoplasmic astrocytes are also going to envelop the synapses, and this is important. And then also they're going to synchronize blood flow with neural activity, and that's important, right? Because if you require more energy and you're processing something that's quite important. For example, in a fight or flight response, you're running away, or you're trying to process a lot of information quickly to ensure your survival. That is going to require more oxygen and more glucose in the brain. And so astrocytes play a huge role in that response, in synchronizing the blood flow to how much activity the neurons require. So how much glucose and oxygen our neurons require. So imagine that if a neuron is trying to process things very quickly but runs out of fuel, you're gonna be slowed down. You're gonna be stopped. Whereas if you have just the right amount of oxygen and fuel, you can actually process things more efficiently. So then we have fibrous astrocytes. They're found in the white matter tracts. They're the least dense compared to the other types, and they envelop nodes of Ranvier. So hopefully you guys remember the nodes of Ranvier are those spaces in between the myelin sheaths. And they regulate the blood-brain barrier. So they're going to help maintain the metabolic and ionic homeostasis. Okay, so basically they're going to regulate metabolism, again, via blood flow and oxygen and glucose, as well as those ions that are required for that metabolism, as well as action potentials. Okay. Interlaminar astrocytes. They're going to move between lamina. So interlaminar, it gets it from the name, using vertical cable-like processes. So in this case, their processes are oriented in a specific way. And their soma, again, soma means body, so the body of the cell, originates in layer 1, but the vertical processes that they have are in the layers 3 and 4. So they can really extend those processes, hence interlaminar. Okay. Varicose. So similar to the interlaminar with long foot processes, but they reside specifically in layers 5 and 6. So those last two layers. Okay, so some of these astrocytes are dependent on the layers of the cortex, and we'll be making a video on that as well. Okay, so the current hypothesis is that interlaminar and varicose astrocytes are essential for higher order cognition due to connections between different layers. So in order for us to process things more efficiently and to modulate how we learn and how much adaptation our neurons have, and our ability, our neuroplasticity is highly dependent on our astrocytes. And so astrocytes play a huge role in that as well. So many people think, and many scientific studies suggest, that these interlaminar and varicose astrocytes that basically interconnect layers of the cortex play a huge role in our ability to learn more complex things. Okay, so how do astrocytes differ from neurons? Right? We know that neurons are excitatory cells. They're going to conduct action potentials. Right? So both astrocytes and neurons express potassium and sodium channels and can evoke inward currents. Okay, this is different from action potentials, right? Because astrocytes do not propagate action potentials along their processes. That is not their function. They're not sending electrical synapses to different cells per se. Now, at least not in the same way as a neuron. So neurons have an all or nothing response. Remember that an action potential has to reach a certain threshold for it to actually propagate, whereas astrocytes have a graded response to stimuli. And that graded response is really important because, again, that helps in how much something is stimulated, how much is not, how much of a reaction occurs, how much metabolism it has, etc. So they're excited by increased intracellular calcium levels. If you remember, calcium is also really important for long-term potentiation, which is how we form memories. So astrocytes probably play a huge role in our ability to efficiently form memories and make memories stronger and weaker. Okay. So here's an overview of normal function. So here I included a picture on the left that shows healthy astrocyte function and also what we call reactive astrocytes. So healthy astrocytes play a huge role in our development. Astrocytes are going to influence the migration of neurons within an embryo. And studies have shown that any abnormalities in this migration can lead to conditions like epilepsy or learning disabilities. Regulation of blood flow is something that we already touched on. So in a healthy person, they're going to help regulate blood flow. For example, if someone's exercising versus sleeping, those require two different amounts of metabolic activity in the brain. Interstitial fluid homeostasis. So they're going to regulate the fluid inside the brain and the ion components inside the brain as well, which is largely going to affect the ability for neurons to propagate action potentials. They're also going to influence on synaptic function. So like I said, they can influence how much neurotransmitter goes in and stays in, and how much of a stimulus the postsynaptic neuron receives, and how that can change the protein expression, which can increase the sensitivity of the postsynaptic neuron. Okay, so those play a huge role in learning and influencing mechanistic shifts. Energy metabolism again, and then obviously the blood-brain barrier. So when we have a reactive astrocyte, which basically just means something that is reactive in a negative way, a lot of bad things are going to happen. So we're going to talk about that. Some of these things are like oxidative stress. They're not going to be able to remove free radical oxygen species. They're also going to change shape, so they're going to change morphology, and we're going to see that in one of the studies later on, okay? So here are some roles in development. So radial astrocytes serve as scaffolding to guide the migrating developing axons. So scaffolding is basically like a format of structure so that things can be directed in the proper way. So you can think of if you've ever passed by a building that was under construction, you can see a lot of scaffolding on the outside to allow workers to get up to higher levels to continue building. Scaffolding is kind of like that. Or scaffolding in the brain kind of serves that way. It directs neurites in certain directions. They can send signals to lure neurons in specific areas and propagate them in certain ways. And so they play a huge role in that. They're also going to release factors such as thrombospondin, which is essential for synapse development. Okay, so again, astrocytes play a huge role in the connections of presynaptic to postsynaptic neurons. They're going to induce the expression of complement system to influence the developmental pruning. So C1q is a protein that is associated with something in our immune system called the complement pathway. So if you're not familiar with that, let us know in the comment section down below, and we'll make a video on that as well. Although if you are eager, you can simply look up complement pathway immunology, and I'm sure there's resources on YouTube or online for that. And they're going to be essential for the development of the white matter, which again, astrocytes play a huge role in the white matter and the gray matter as well. So regulation of blood flow. Astrocytes are going to release a lot of molecules that are going to help mediate these processes. For example, prostaglandins, nitric oxide, arachidonic acid. And these factors will help increase or decrease blood vessel diameter and thus the amount of cerebral blood flow, right? So if you increase the vessel diameter, you may be increasing the blood flow. If you decrease the blood vessel diameter, you would be increasing the pressure inside the brain as well, the vessel pressure. So those two things have different effects in our physiology. And blood flow is closely modified based on neuronal activity due to connections of vessels and synapses mediated by astrocytes. So for example, if we go back to that picture, so right here, if this synapse is highly active in some pathway, the astrocyte that is communicating with the synapse can also respond to this via the blood vessel. And so because of these interconnections and connectivities of the astrocytes, where it's also modulating the communications between neurons, but also modulating the blood vessel. This astrocyte can regulate the flow of blood here, as well as the influx of glucose and oxygen to influence the metabolism of these neurons, which are highly active. Okay, so neurotransmitters, neurotransmitter homeostasis at synaptic cleft. Again, so the astrocytes are going to regulate that synapse, which are going to influence how much neurotransmitter is there, how much is left, how much is uptaken, how much is left there. Okay. Networks of astrocytes via gap junctions rapidly dissipate accumulation of molecules to prevent potential accumulation. So they also help clean up synapses. Okay, so this is highly important because if things remain in the way of synapses, they can potentially block the stimulus that's required for the postsynaptic neuron to that the postsynaptic neuron is ready to get. So anytime we have something in the way of a synapse, that can influence the postsynaptic response. And so the astrocytes play a huge role in that as well. So astrocytes are also going to regulate the release of these active molecules here. So glutamate, purines, GABA, and D-serine. So we have a couple of amino acids. GABA, again, a really important neurotransmitter. Glutamate as well, very important neurotransmitters. And these are usually in response to astrocytes being activated via an increase in calcium 2+ concentration, or calcium ion concentrations. Okay. So tripartite synapse is a hypothesis stating that astrocytes play a direct and interactive roles in synaptic activity essential for information processing by neuronal circuits. So we can't just have a large circuitry of neurons, we also have to have astrocytes regulating these processes in different ways. So astrocytes are also going to influence the synapse by secreting certain growth factors. And these growth factors are important for the remodeling of the adult brain. So this is what allows us to be plastic, which is what I've been saying throughout this presentation. They're also going to release factors like TNF-alpha, which is going to induce expressions of AMPA. TNF-alpha is one of those inflammatory cytokines, stands for tumor necrosis factor alpha, and it's going to be important in something called gliosis, which we'll talk about in a second. And they're also going to release some steroids which modulate GABA receptors. So how an astrocyte reacts to its environment is directly going to influence the presynaptic and postsynaptic neurons and how they're going to communicate. So astrocytes, like I've said before, they're going to take up glucose from the bloodstream and they can store it as glycogen, which glycogen will then be broken down into glucose to give to the neurons when it is needed. And astrocytes, astrocytic glycogen stores are used in hypoglycemia or high neuronal activity. So basically, if you have a lot of neurons firing that require more glucose to maintain that functionality, that higher function, you're going to need a larger amount of glucose stores. Now, there are a lot of old psychology studies that looked at how much a person can concentrate on a single subject. So imagine you're taking a test, right? And there's a question. And what they found was that their ability to concentrate was directly related to, or at least directly correlated and postulated that it was directly caused by how much glucose they had in their blood. So when a person's thinking a lot and they haven't had a lot of food, the glucose in their blood goes down, so their blood sugar goes down, meaning that their neuronal activity is also going to have to go down because there isn't enough fuel to fuel the neurons that need to be firing at that rate. And so one of the things that everybody suggests before you take an exam is to have a well-balanced and decent meal so that you don't lose focus without. And if you take large, long exams like an MCAT, the medical school's admissions test, or an LSAT, or any large test that takes multiple hours to complete in a single day, they give you food breaks. So that, and there's a reason for that, right? You can't just fast the entire time and do it. You might not have the same performance. So astrocytes play a role in feeding the neurons, but whether it just be delivering glucose directly from the blood or storing excess glucose to feed them when times get rough, right? And then glucose metabolites such as lactate can be transmitted through the gap junctions in a process which is glutamate regulated to synapses in gray matter and nodes of NVA in white matter. So again, the astrocytes, how they're going to play a role in the feeding of the neuron depends on where they're located, whether or not they're in the white matter or in the gray matter, and directly dependent on the neuronal activity and blood glucose levels. So here's an interesting picture that shows the tight junctions of the blood cells, or of the blood vessels where the blood cells are going to pass through, and the astrocyte processes that kind of coat this blood vessel. Okay. So these feet are going to impede the molecular influx into the brain based on the polarity. Also, the blood-brain barrier will allow things to pass based on size. But there's a lot of things that go into what can and can't pass through the blood-brain barrier readily. And it's one of the challenges in drug delivery into the brain. So let's say you design a drug that works really well in saving the lives of neurons in a dish. That doesn't necessarily mean that if you administer that drug into the patient orally or intravenously, that drug will make it to the blood, through the blood-brain barrier and into the brain where it's needed. So whenever you're designing a drug, or for example for Alzheimer's, you have to make sure that drug can readily pass the blood-brain barrier to do what it's intended to do. Okay. Blood-brain barrier leaks occur when astrocytic feet or astrocyte feet have dysfunctions or deformities because of certain mechanisms, molecular mechanisms. So this is what I was talking about, gliosis, right? So if there is an injury to the brain, there's going to be what's called reactive gliosis, where there's a process of inflammatory markers that are released either by astrocytes or by microglia. Oftentimes it's by microglia. And those inflammatory markers, inflammatory cytokines like TNF alpha or interleukin 1 alpha, are going to cause morphological changes in the astrocytes and surrounding cells, which may reduce the feed processes that astrocytes have. And so this is important because if you diminish or change the morphology of the astrocyte, you are therefore impeding its function or changing its ability to function. So morphology has a direct relationship to the cell's ability to function. And that's why this gliosis, reactive gliosis, is important to study. Okay. However, this note here is important as well. Astrocyte regulation is debated because there is in utero evidence of BBB, or blood-brain barrier regulation, even before the formation of astrocytes. So there may be other roles that are playing, or other things at play here that we haven't fully disclosed yet or understood yet. But it's safe to say that astrocytes do play some role in the blood-brain barrier, and simply that we need to study this more closely. So what is reactive gliosis? Here's the textbook definition. So gliosis is a non-specific reactive change of glial cells in response to damage to the central nervous system. And this can be blunt to the head, it can be a stroke, it can be anything internal or anything that's caused externally, like a traffic accident, for example. In most cases, gliosis involves the proliferation or hypertrophy of several different types of glial cells, including astrocytes, but also including microglia and oligodendrocytes. Okay. So proliferation, what does that mean, right? Proliferation just means that these cells are going to be multiplying. So you start off with, one cell, then you get 2, then you get 4, then, et cetera, et cetera. So they're going to proliferate or increase in numbers, and they're going to potentially hypertrophy. And these mechanisms are again, not perfect. They're debated. It depends on the scenario, depends on the cell. Sometimes what is also sometimes noticed is that they will hypertrophy, and then after some time, they will actually shrink. They'll actually atrophy a little bit. So I guess it depends on the time, but it also depends on the cell. But for those of you that don't know, hypertrophy is the increased cross-sectional area. Basically, it means to get larger. So it means that the cells are swelling. You can think of it like that. And atrophy would be the opposite. Atrophy would mean that you are losing cross-sectional area, that you are shrinking. You can think of this as in cell size, or you can think of it, as a personal trainer, we talk about hypertrophy in terms of muscles. So if you want to get bigger muscles, it's called hypertrophy of the muscles. So think of it that way. Hypertrophy is getting larger, and proliferation is making more, multiplying, right? Technically dividing, but whatever. So glial scarring. So as astrocytes proliferate, let's say that there was a stroke, right? And those an ischemic stroke, a dry stroke, and those cells that were in close proximity to where the blood vessel closed, they're going to die because they don't have any oxygen or nutrients coming in. So those cells are going to atrophy, and eventually they're going to go through the apoptotic pathway. They're going to get basically eaten up and recycled, and they will no longer function. And so because of that, because they essentially dissolve away in a sense, that space is going to have to be taken up. And so what happens is that these astrocytes, running astrocytes, are going to change their morphology, they're going to proliferate, and they're going to take up that space, creating a scar. You can think of just how your skin scars, but instead of your skin, you're talking about the brain, and the scar tissue is made up mostly of astrocytes here. Okay, they're the main cellular component of the glial scar. And this is especially important if you're taking out a tumor, right? If you're going to resect a tumor from someone's brain as a neurosurgeon, you have to consider the scarring that's going to happen afterwards. And so there are huge enterprises of research looking into how we can minimize glial scarring and actually induce proliferation of more neurons to take up the place of those neurons that have died. Although implanting new neuronal cells, again, won't bring back the function, but potentially it can increase the potential for how much function the person will have after some training, right? Because if you lose the neurons that you have, your neurons are unique to you and your experiences and your skills and your abilities that you've acquired. So when they die, they're gone. You're going to have to rebuild those and rebuild those synapses in a new and different way. But you can only do that if there are neurons available for that process. So that's one of the realms of research. Okay, so after some type of injury, whether it's a blunt force to the head, like a TBI, or a knife wound, a gunshot wound, or a tumor, whatever it may be that injures your brain, astrocytes are going to form a dense web of their plasma membranes that are going to fill up that empty space, and that's called the glial scar. And this is going to modify the extracellular matrix, which is basically the stuff that's outside of the cells, where the cells are kind of sitting. And so they're going to, astrocytes and other surrounding cells are going to secrete molecules that unfortunately inhibit neuronal growth and regeneration. So that's another barrier is making sure that we can increase the proliferation of neurons, whether it be in a natural way through a drug or through implantation of stem cells, because the natural mechanism of our brain after injury is to inhibit that neuronal growth or regeneration. Okay, so these are some molecular triggers that we know cause glial scarring or reactive gliosis, basically changes in the morphology of the cells that can cause disease states. TNF alpha, IL-1 alpha, and IL-6, those are cytokines that are secreted by, for example, microglia. And when those cytokines reach astrocytes, they're going to cause changes in the morphology. And when you change the morphology of an astrocyte, you change the function. And if you change the function or diminish the function of the astrocyte, you're diminishing the regulation of synapses. And that can mean that there's no policing of the synapse from secreting too much neurotransmitter such as glutamate, which can be toxic, right? So you're not keeping things in check, you're not keeping things in balance and homeostasis, and so negative things are going to happen. Too much upregulation of certain things, too much excitotoxicity in certain areas, and so it just becomes a mess, an unregulated mess, and things start to break down because of it. So here is some figures from a research article that's cited below. Aberrant iPSC-derived human astrocytes in Alzheimer's disease from Cell, Death and Disease. And what they looked at were human-derived astrocytes, both from healthy wild-type patients, so patients that were healthy. They also looked at people with familial Alzheimer's disease and sporadic Alzheimer's disease. So FAD stands for familial Alzheimer's disease, which means the person has a genetic predisposition. It runs in their family. And then sporadic Alzheimer's disease is just a naturally occurring, there's no real molecular or genetic factor from family, it just occurred. And what they found was that healthy astrocytes have a completely different morphology. They have more branching, which is where the name aberrant comes from. It means tree-like. And then familial and sporadic Alzheimer's disease derived astrocytes have fewer processes, if not any. And the paper noted that they look fibroblast-like. So they're very round. They don't have expression of this S100B protein, which is characteristic of healthy adult astrocytes. And so you can see those morphological changes if you look down here. These are 3D scans based on what was found here in the confocal microscope or confocal imaging. And the familial and sporadic Alzheimer's disease, these are astrocytes. Notice how flat, and there's no branching. So you'll never have that branching that we saw back here where those processes are sticking out towards the blood vessels and towards the synapse. And so those things, basically, it's like all those branches were cut off, and you were only left with the body and the nucleus. And so that change in morphology changes the function. It means that there's going to be a lack of function. And here's the healthy astrocytes in a three-dimensional diagram. Okay. And here are some of the graphs. So here again is another showing the branching of a healthy astrocyte versus the fewer branching, the smaller volume, smaller surface area of the sick or unhealthy astrocytes. And here are various graphs describing their volume, the average volume of the healthy compared to familial, compared to sporadic, the average surface area to volume ratio of each, which is important for cells and cell function. Notice that the healthy astrocytes are significantly outperform everything besides the GFAP, which is another characteristic protein of astrocytes. So you can tell that these are astrocytes, but there was no significant change of the glial fibrillary acidic protein. However, there was a lot of change in the S100B and other proteins that are known to be expressed by healthy adult astrocytes. So this was pretty interesting. Okay, so here's a summary of everything that we talked about. So astrocytes have many functions in the central nervous system, including they have roles in development, they regulate the blood flow, they regulate the interstitial fluid, they influence the synapses of neurons, they have huge roles in energy and metabolism and the blood-brain barrier. There are 4 classes of astrocytes that you guys should be familiar with: interlaminar, protoplasmic, varicose, and fibrous. And astrocytes are known to play a role in higher cognition as well as disease states such as Alzheimer's, ALS, etc. So they're critical for normal, healthy, higher cognitive function that we have as humans, but they can also, if they change in their morphology or after disease, after injury, they can be limiting in their ability to allow us to regenerate neurons. So it's a double-edged sword there, I guess. Okay, and here are 2 final images to show you guys. This is a phase image. These are from Science Online. They sell human astrocytes, so these are some of their images. This is a phase contrast at 100 times. You can see all of these are individual astrocytes kind of branching together, and this is in a dish in vitro. And then here they are in a confocal setting where they've been stained and fixed. And I believe, I don't know for sure, but I believe this is GFAP stained, and then DAPI. DAPI would be the nucleus, and then GFAP would be characteristic protein. Might be S100B, but it looks more like GFAP to me. So the glial fibrillary acidic protein. But that's all we have for you today. If you guys have any questions, put it down in the comments, and stay tuned for more videos.